Okay, so hello everyone. Oumaima and myself are research engineer at the group Decision and Bayesian Computation at the Institute Pasteur. So we are going to present you the software DIVA for image treatment using virtual reality. So first, the 3D image obtained by microscopy can be really vast, huge, and not standardized because of, for example, the biology of the element of interest. It can also depend on the protocols used to prepare this sample, so there is a kind of a human factor. And finally, it can depend on the hardware used to capture the image, because with some microscope, you can have really vast and huge information on the image, and some microscope needs some complex parameterization and then requires some specialists to help you to fix with this parameterization. So with this 3D complex image and vast image, you need to extract some meaningful information, which is the goal of image processing. And in order to do that, you need to define some image features. So for the image features, you can define some uncrafted features, for example, through, through macros of Imabio, for example. And even if this method is really fast, it has an uncertainty intolerance. So a more robust solution is to define learned features through, for example, a neural network or decision tree. And this method is statistically more robust than the previous one. But there is a huge pitfall because you need a massive amount of data to train this network. And there is a kind of black box contained in the hidden neuron layer because you don't know exactly what's happened on this layer and you cannot control their behavior. So a third solution is to define some contextual features which are adapted to the user context and thus need the user input. So for these contextual features, you need a human interaction. And for the human interaction, the visualization is a key. In the laboratory, we choose virtual reality for this visualization because it combines a stereoscopic view. So stereoscopy is the fact that you have two images in front of you with different positional and rotational offsets. And thanks to the stereoscopic view, you have a total immersion in the virtual environment. So your object of element, your volume, looks like a physical object in front of you, and you can directly interact with this object thanks to the motion tracking of virtual reality. So in the laboratory, we have gathered three aspects of the data, the contextual intelligence, and the user interaction in order to create a human-in-the-loop solution named DIVA for data integration and visualization in augmented and virtual environments. So now, how DIVA works. So I will show you here an example of a mouse hippocampus. Sorry. So you can see this picture in Imabio. So it looks like a zit stack. You know how to use DIVA. So you just need to open the software here, and then you can drag and drop the file on the software. And DIVA will read this Z stack image as a volume, and you can see the volume here. So for now, you have just desktop view, a 2D view of the software, and you can directly interact with the volume. So after loading the volume, you can control some visualization parameters to see just your element of interest. For example, here the user wants to disable the background. You want to disable the voxel, the black voxel, for example, here. So when your visualization parameters looks fine for you, you can directly switch from the 2D view to the 3D view with the virtual reality. So for that, you just need to use the headset. So put the headset on your head and take the VR controller. So when you use the headset, you will see your element, your image, as a physical volume in front of you, physical object. And with the controller, you can directly interact with it. You can bring the volume closer. You can move around, you can turn the volume. You can also, with a clipper object, go in and out of the volume. With the controller, you can do some different measurements in virtual reality. So for example, here, the user wants to count some the nuclei, for example, for this image. So in 3D, it's just a point on the nuclei and add some points. And then in a 2D, you can extract the number of points. You can extract the coordinate of this point. You can also, I don't know, calculate the distance between 2 points or calculate the volume of an element in your image. So thanks to the headset and the controller, it is really easier and faster to visualize some complex 3D image, to interact with them, and also to make some measurements that can be really difficult to carry out with the 2D software such as Imabio, for example.
And here we would like to talk about a recent project mixing DIVA, virtual reality, and neuronal cell development. This research project is conducted in collaboration with the SPEDER lab, and we are working on the analysis of the clonal development of Drosophila larvae in brain. What we would like to determine is whether the organization of clones in the brain follows certain rules. For example, is there a constant number of neighbors in 3D? Is there a correlation between clone size and ability to overlap? Is there a constant number of clones per region of the brain? Actually, the ultimate goal for the biologist is to open the way to understand the mechanism behind these rules. And to do so, we need to classify the clone according to their anatomical criteria. But the first step before starting the clone analysis is to extract each clone from the original image. At the beginning, we thought about fixing some clone criteria such as density, texture, etc. Then extract the clone automatically from the image. But as you can see in the following picture, we show 5 samples of Drosophila larva brain with different size, shape, orientation, and density. So standardize this database could be quite difficult and challenging. In this slide, we show another sample where we decompose the image to 4 channels. Here, I just want to show that except channel 2, where the clone are where the clone are visualized, we have low contrast images and super weak signal of the clone in high intensity noise, which prevent us from automatically detecting borders. Thus, we need manual tagging using DIVA. DIVA provides more realistic images of the larva brain, which make a clone easily interpreted by anyone, even a non-biologist. And in this video, I will explain briefly how I tag a clone. I open the brain image in DIVA. I keep the channel that I need to extract its clone and exclude the others. I display the interactive curve of the one-dimensional transfer function. And by manipulating this transfer function, I define visually the clone. Then I start the virtual reality vision. Here I choose the selector tool. It's one of the tools that we have in DIVA. And then I start tagging. The additional dimension given in virtual reality allow us to have more views of clonal structures that were not available in 2D application like ImageG, for example. I want to mention that I had a difficulty with some samples to estimate whether 2 clones are connected or not. Some signals are too faint in parts, and the clone seems like having overlapping contours. And in most of the case, we needed the intervention of the expert biologist. After finishing the manual tagging with DIVA, we obtain for each image 4 images of mask corresponding to the 4 channels that we have. Then we start the post-processing on the generated mask. So we apply the watershed segmentation algorithm on each channel. This algorithm is applied to affect an intensity value to each clone. So the max intensity indicates exactly the number of clones in the current channels. in the current channel. So to extract each clone, we just set a threshold value on each intensity, and our output is a stack of image with the clone mask volume, and this mask is multiplied by the original image of the brain to obtain the corresponding clone image. The last image on the right of the slide shows each clone with different color. Since we have all clone volumes separated, we extract physical properties such as eigenvector matrix, eigenvalues, inertia tensor matrix to define the orientation, for example. And we extracted also the morphological criteria such as volume and surface. And all these information serve to compute the distance between border of each clone in the same channel. We are talking about the inter-class neighborhood. The distance between border of each channel in different the distance between border of each clone in different channel, and we are talking about inter-class neighbors, and the volume between clone that overlaps. Another problem we need to solve is whether the clone is in the central brain lobes or in the ventral nerve cord. And to answer this question, we extract the larva brain skeleton by denoising the third channel with a Gaussian blur. Then we apply a 3D skeletization algorithm. And for each clone, we project the centroid coordinate, the 2D centroid coordinate of the skeleton image, and we compute its distance compared to the branch. Our perspective or our future work is to answer two questions. The first question is there a correlation between clone size and ability to overlap? And the second question is there a constant number of clones per region of the brain?
Okay, so now the second possible application with DIVA is in the medical field. So here I will present you an example of an heart disease called criss-cross congenital defect. And this is a project in collaboration with the Dr. Francesca Remoni from the Necker Hospital. So now in this video, so you can see the heart as a physical object in front of you. And then the user can directly interact with it. So with the controller and the clipper tool, you can go in and out of the heart. And the goal is to visualize and understand the inside and outside structure of the heart. And in addition to the 3D view, you can see here on the box some 2D projection, which corresponds to the projection of the volume at the coordinates of the controller. So you both have the 2D visualization and the 3D visualization. So all these different applications in the medical field or in microscopy are led to 5 different publications linked with DIVA. And the software DIVA is a project of Mohamed El Beherri, Jean-Baptiste Masson, and Maxime Daron. And we also want to thank all our different collaborators in the different institutes, so in the Institut Pasteur, Institut Curie, the Necker Hospital, the Paris University, or NVIDIA, and all our different sponsors. In addition, you can try DIVA with your own data and try to see what you want to see in the virtual reality by downloading the software and the user manual at the website diva.pasteur.fr. Attention.